Very glad to open this session about vascular factor in Alzheimer and to show you our results on how cardiovascular and cerebrovascular disease promote Alzheimer pathological changes in preclinical Alzheimer disease. And I have nothing to disclose. So I think it's quite clear in the literature and also from the latest Alzheimer criteria that vascular pathology and Alzheimer disease often coexist and present together in patients. And there seems to be a specific relationship between cardiovascular and cerebrovascular disease with amyloid deposition, so the early stages of Alzheimer. However, I think there are two main questions that are still open in the literature. And the first one is about whether this relationship is merely due to shared risk factors, such as, for example, age, primary age, but also genetic factors such as ApoE4, or whether there is an intrinsic and mechanistic relationship between these two diseases. And the second main question that is still open is about the promotion of downstream AD biomarkers by cerebrovascular and amyloid, and specifically whether they would contribute to this, to p-tau atrophy and cognitive decline in an independent or in an interdependent synergistic way. So in this project, what we wanted to do was to answer these two main questions, and we started with first describing the association between vascular risk factor, cerebral small vessel disease, and Alzheimer's disease CSF pathology in non-demented individuals. And then we wanted to evaluate whether vascular risk factor promotes amyloid pathology and whether this goes through the presence of brain vascular lesions. And eventually, we wanted to model the whole influence of vascular factor in the amyloid cascade of events. So to do so, we used the European Prevention of Alzheimer's Dementia cohort. And by the way, all the data that I'm going to show to you today are now available online and can be accessed through the website. And the EPAD cohort targets the early stages of the disease and includes individuals above the age of 50 and without diagnosis of dementia. We use CSF biomarkers of Alzheimer, which are amyloid 42 and p-tau 181. We measured atrophy with MRI, and specifically we performed free surfer to compute volumes within an AD signature mask. And we measured cardiovascular risk factor using the Framingham risk score, which is a general composite score of various cardiovascular factors. Importantly, we had a broad cerebral small vessel disease radiological evaluation, which I'm going to talk about in the next slides. And eventually, we also have cognitive information, so cognitive performance with the R-bands test, which is a test about global cognition. But we're currently also exploring specific domains, but I'm not going to show these results today. So, as promised about the cerebrovascular burden radiological evaluation, we had the FASICA score, which is a 0 to 3 scale for white matter lesion, and we specifically had it divided in periventricular and deep lesions. We also had information about microbleeds, and we stratified our participants as having less than 2 and more than 2 microbleeds, and we also had information about whether they were deep or lobar. We had information about lacunes, and we stratified participants having 0, 1, 2, or more than 2 lacunes. And eventually, we also had a scale about enlarged perivascular spaces, which is a 0 to 4 scale. And in this case, we had also information about whether there was in the centrum semiovale, so the upper part of the brain, or in the basal ganglia. Aside from this radiological information, we also had quantitative information because we ran what is called Bayesian model selection, which is an automated segmentation pipeline for white matter lesions, and we obtained white matter volumes in 8 regions of interest, which are the 4 lobes, again divided by periventricular and deep lesions. So in this slide, you see the cohort characteristics. Generally, we ended up with 1,592 participants, so quite a broad cohort for these early stages. You can see that most of them are in the A-T+, but we also have some positivity. Importantly, age, as expected, was around 65, and even more importantly, the Framingham score was around 14, which shows a low to intermediate cardiovascular risk. And this was also confirmed with the cerebrovascular indices, which I don't have in this table because it's too big, but also there we saw a low to intermediate vascular burden overall. So let's start with our aim, and I'm going to show you the statistical analysis and the results that we have. And the first aim was a descriptive one. So we just wanted to look at the general association within vascular risk factor, cerebral small vessel disease, and Alzheimer's disease CSF markers. And to do so, we used linear models. And we had as an outcome the CSF markers, so first amyloid and then tau. And we used Framingham and each of the CSVD radiological indices in separate models to study this association. And importantly, when predicting tau, we also added an interaction between the CSVD markers and amyloid to study and to look for those interdependent effects that we mentioned before. So the first thing that we found that higher cardiovascular risk factors were related to both CSF markers. And here you can see that higher Framingham was related to lower beta-42, so more amyloid pathology, but also to higher p-tau in the CSF, even if this last association was strongly impacted by the correction for age. But most importantly, we found that CSVD burden and all the CSVD indices that we look at were related to amyloid pathology. So basically, when looking at perivascular spaces, as you can see with the last two groups, but also when looking at lacunes, as you can see with only the one having more than 2 lacunes, and microbleeds, specifically for the lobar microbleeds as expected, and eventually also and more strongly for when-measured lesion, both radiological evaluation and quantitative evaluation, we found that having a higher cerebrovascular burden was related to lower levels of amyloid beta-42 in the CSF. So again, more pathology. When looking at tau, we found a significant interaction between some of our CSVD indices and amyloid in predicting tau, and specifically for the two when-measured lesion indications, so the fascicles and the volumes, but also for microbleeds, we show that having a higher cerebrovascular burden promotes a stronger relationship between amyloid and tau, so more negative relationship in this sense. So again, advocating for an interdependent effect on p-tau. So given this first descriptive analysis, we wanted to go further, and we wanted to model whether vascular risk factor promotes amyloid pathology and whether this is mediated by vascular brain lesion. So to do so, we use structural equation modeling because this allowed us to use confirmatory factor analysis to measure one latent factor that we call the CSVD severity latent factor, which is basically a principal component obtained by all our radiological indices that I've shown you in the previous slides. And using SAM, we could model whether this latent factor was mediating the association between Framingham and amyloid that we saw in the previous descriptive analysis. So first of all, here you see the distribution of this latent factor across our cohort, and you can also see the contribution of each of the indices. And as you can see, the fascicle scores were the one mostly contributing to the CSVD severity, followed up by the perivascular spaces, and eventually lacunes and microbleeds. But most importantly, we observed that the association between Framingham and amyloid that we saw before was fully mediated by the CSVD severity latent factor. So all that association was due in our cohort by the presence, was going through the presence of vascular lesions in the brain. So we got to our aim 3, which was to model this whole influence of both cardiovascular and cerebrovascular pathology in the whole amyloid cascade of events. And again, we used SAM, but we expanded a little bit the model, and I'm going to show you what this means. So on the left part of this model, you see exactly the same mediation that we have seen in the previous slides. So Framingham, CSVD severity, and amyloid. But this time, we added those downstream biomarkers that we've been talking about, so CSF p-tau, atrophy, and cognitive performance. And we studied the direct and indirect effect of both amyloid and CSVD severity on all these biomarkers. So in this slide, you see the results. I know it's quite a lot, so we can go through it together. So easily, on the left part is exactly the same mediation that you've seen in the previous slide. So CSVD severity mediating the effect of Framingham on a beta. But if we focus on the right part and we look at the red, so the direct effect, the red lines, first we see that amyloid directly impacts tau, atrophy, and cognitive performance, and this was expected, but also that CSVD severity directly impacts the atrophy measures and cognitive performance. But probably the most interesting results are the blue lines here, which shows our indirect mediated effects. And as you can see, we found that CSVD severity latent factor has a significant impact on all the downstream biomarkers mediated by amyloid. So it can accelerate the amyloid cascade, but this is fully mediated by the presence of amyloid in our case, or partially mediated for some of them. And the last result I want to show you, we tried to expand this model with longitudinal data, which we're still working on. This is a very recent result. We managed to have some longitudinal data for only the last two endpoints here, so the atrophy and the cognitive performance, and we inserted rates of change in the models. Of course, our sample goes down quite a lot with still 456, and the follow-up is not very long. But as you can see with the blue line, we confirmed this association with the atrophy, while we didn't find anything with cognition, but this is expected considering non-demented individuals, a very short follow-up. So with that, I want to conclude. We further confirm previous finding, but we also expand on the detailed radiological evaluation which is present in EPAD, and we show that vascular risk factor promote amyloid fully mediated by the presence of vascular lesions, and eventually that CSVD promote downstream AD biomarkers through amyloid pathology. With this, I thank you for your attention. I want to thank all the co-authors who have been really involved in this work. A special mention to Alessio, which has been suffering with me on this work a lot. So thanks for that, and thanks to you for your attention.
It's my pleasure to introduce Teresa Harrison, who's going to give the next talk on the impact of Alzheimer's disease pathology and cardiovascular risk factors on cross-sectional brain structure in the U.S. Pointer imaging study.
Thank you. I have no disclosures. So models of AD pathogenesis have been largely informed by biomarkers, as we all see at this meeting, and the associations between them. But these associations have mostly been explored in homogeneous cohorts, and homogeneity or heterogeneity can be measured across many different dimensions, including ancestry, race, education, cardiovascular risk, and other health risk factors. And biomarker relationships may vary according to cohort heterogeneity. So in this study, I'm going to look at two different cohorts that are very different in their characteristics. First, the Pointer imaging cohort, which is a community-based sample recruited based on cardiovascular risk, racial and ethnic diversity, and other risk factors, and compare it to ADNI, which is a clinic-based sample with relatively less recruitment emphasis on risk and diversity. And the dimension of heterogeneity I'm going to focus on today is cardiovascular risk, which we know is related to cognitive decline and a higher risk of conversion to dementia. But there is relatively less evidence linking CVD to AD pathology directly. And as a little bit of background, I'm going to share a little bit of some work from Susan Landau, who's also comparing these cohorts, and has found that the interaction between amyloid and tau that we see in many cohorts, or has been seen in many cohorts, is relatively less pronounced in Pointer, such that amyloid is still the screen. So given this, we hypothesized that perhaps interrelationships between cardiovascular risk, brain structure, and cognition may be stronger in Pointer compared to ADNI and represent a non-AD pathology pathway to cognitive decline, because these two cohorts are actually similar in their cognition. So this is some information about the particular participants I'll be looking at today. The ADNI group was older, but in terms of sex and A-beta status, the groups were not significantly different. And I'll be sharing some key harmonized variables and the relationships between them, namely the T1 MRIs we process identically for VBM analyses, which will be the focus of the talk. We calculated a preclinical Alzheimer's cognitive composite score for both cohorts. And we also calculated Framingham risk score, which you just heard a little bit about, which represents the 10-year percentage risk for a major cardiovascular event. And the components of this score are age, sex, diabetes, smoking, systolic blood pressure, and body mass index. We didn't have lipid data available for ADNI, so we went with the alternative of using body mass index. And as you would expect, based on the recruitment emphasis is significantly higher in Pointer than in ADNI. We also calculated cortical summary burden in centiloids and tau burden in the entorhinal cortex. And while we did employ identical processing and quantification, the two cohorts used two different tau tracers, MK in Pointer and FTP in ADNI, and we don't have a harmonized scale, so we can't directly compare them. So looking at a few of the relationships between some of these variables, we see that there is this association between FRS and cognition in Pointer and in ADNI. But if we look at the relationship between FRS and amyloid burden measured with PET, obviously, and entorhinal tau. We don't see a relationship in Pointer, and we don't see a relationship in ADNI either for the relationship between FRS and amyloid, and there is a small effect size relationship between FRS and entorhinal tau. So given that AD pathology is not related or weakly related to FRS and is less related to cognition in US Pointer, as I showed you in Susan's work, what is the role of cardiovascular risk in the brain, in the well-understood or well-established brain structure cognition relationships, and does that vary by cohort? So I'm going to show a lot of these surface renderings. So before I get into these specific results, I'll just want to orient you to the display. So all the color bars you're going to see are the same, and the bottom of the color bar represents a statistical threshold of p.001 uncorrected, and just shy of the top of the color bar is FWEP less than 0.05. So anything you see in yellow in these maps is statistically significant after correction for multiple comparisons. So this particular analysis shows the positive association between the cognition measure, PAC, and gray matter across the brain, and we see these nice familiar regions for many of us in the temporal lobe and the posterior medial parietal lobe. And in ADNI, we also see temporal regions and similar cortical regions, but we wanted to know, of course, well, this is what it looks like side by side. What does a cohort by PAC interaction in a single model look like? And we found that there are regions where the PAC gray matter association is stronger in Pointer than in ADNI, and we didn't see anything in the opposite comparison. And if we take the mean volume of these regions, we see that there's a partial significant mediation of the FRS between this in this gray matter PAC relationship. This mediation is significant for all 1,050 participants and per Pointer participants, but not ADNI participants alone. But this is really just the beginning of this exploring this relationship. Because there are other methods to establish regions that are associated to cognition and see if this FRS relationship holds. Next, we're going to shift to looking at the relationship between entorhinal tau and brain structure. And you see here in Pointer, there's this nice, strong association between entorhinal tau and structure in the medial temporal lobe, and you can see it even more nicely in these slices. And in ADNI, we also see a medial temporal relationship and some additional cortical regions. And I just want to, we did not find any significant gray matter associations between looking at any amyloid measure, so either amyloid continuously or an interaction between amyloid and tau. I just want to quickly point out this very interesting finding that replicated across both cohorts, showing the relationship between entorhinal tau and atrophy in the basal forebrain, which is a region I'm very interested in. I'm happy to answer any questions about that even after the talk, if anyone wants to talk more about that. But I'll shift gears to talking just about the final measure that we looked at in relationship to brain structure, and this is FRS, of course. So in Pointer, we found this relationship when we looked at the negative association between FRS and gray matter. So this occipital cluster popped out at us. We are not sure exactly what to make of that yet, but we don't see any association between FRS and brain structure in ADNI. Now, despite the fact that these maps look different, there was no significant cohort by FRS interaction when we put them in a single model. Now, those of you who have done these type of analyses are probably wondering, what about age and sex? Age and sex are components of the FRS score, but they're also huge drivers of brain structure. So we had to covary for age and sex in order to find this pattern. So we had to ask, well, okay, what does this pattern actually reflect? So to do that, we went and dove a little deeper into the components of the FRS. So these are just the same surface renderings I showed you on the last slide. But now we're going to look at body mass index, which is one of the continuous measures included in the FRS. And in Pointer, body mass index was significantly higher. And we found when looking at the negative relationship between BMI and gray matter that it recapitulated the pattern we saw when we looked at FRS in a much more statistically significant way, and also there were additional clusters. And we don't see any meaningful pattern in ADNI. We then looked at systolic blood pressure, which actually was slightly higher in ADNI compared to Pointer, and we don't see much of a pattern in either cohort, which led us to conclude that what we saw when we were looking at this cardiovascular risk score and brain structure was really driven by BMI in this case. So to wrap it up, we found that Pointer and ADNI share similar regional patterns of positive cognition brain structure relationships, but that these associations were stronger in pointer in several key regions, and that the relationship between mean volume in these regions and cognition was partially mediated by FRS. In ADNI, entorhinal tau showed a more regionally widespread effect on cortical volume compared to pointer, where the effects were predominantly restricted to MTL and basal forebrain. And finally, measurements of cardiovascular risk, including BMI, are related to cortical structure in pointer, but not in ADNI. So I just want to note that this is really the tip of the iceberg of what we have to do to look at these associations in these two cohorts and understand the effect of cohort heterogeneity. But I've listed just a few next steps of what is actually many. We're going to explore more formal matching of ADNI participants to pointer participants. We also would like to do some additional harmonization of cognitive measures so we can look at domain-specific composites, and then also perform some path analyses to actually help work out this hypothesis even further, that there may be different pathways to brain atrophy that then synergistically affect cognition. And with that, I will thank the JGIS lab, and many thanks to the participants, sponsors, and staff of both Pointer and ADNI, which are both huge undertakings.
It's my pleasure to introduce the next speaker, Emma Koumans, who's going to talk about the role of cerebrovascular pathology in the association between amyloid and tau.
Thank you for this introduction. Is this working? No? Hello?
No.
It's not working, I think, no?
Try the other one.
Hello?
Yeah.
All right. Thank you for this introduction, and thanks for the organizers of the AIC for the opportunity to speak here. These are some disclosures of my co-authors, but I personally have nothing to disclose. So from previous studies, we know that Alzheimer's disease pathology very frequently coexists with cerebrovascular pathology. And we also know that cerebrovascular pathology can contribute to Alzheimer's disease-related clinical progression. But to what extent cerebrovascular pathology can contribute to Alzheimer's disease pathological progression remains less clear. And findings from the literature are actually quite mixed. So some studies have reported significant interactions between the presence of vascular burden and the presence of amyloid pathology on subsequent tau accumulation, whereas, for example, other studies did not observe any significant associations between vascular burden and tau. And so we felt it was important to investigate this further. So the aim of this study was to investigate whether cerebrovascular pathology interacts with amyloid pathology in the association with both baseline tau and longitudinal tau accumulation. And to this end, we included 1,240 participants from the longitudinal Swedish Biofinder 2 study, who were all 50 years or older. We included amyloid negative and amyloid positive cognitively unimpaired individuals, as well as amyloid negative and amyloid positive cognitively impaired individuals. And just to note is that there were no non-AD dementia cases in the amyloid positive impaired group, so only MCI and AD dementias. All individuals underwent baseline tau PET using the R0948 tracer, and then we quantified temporal meta-ROI SUVR. A subset of 678 individuals also had longitudinal tau PET available with an average follow-up time of about 2.5 years. For amyloid pathology, we had to use different measures of amyloid for the unimpaired and impaired individuals. So for unimpaired individuals, we use a continuous measure of neocortical global amyloid PET. But for impaired individuals, we had to use a dichotomous measure of amyloid status based on either PET or CSF, because amyloid PET was missing per protocol for the vast majority of dementia cases. We focused on three main cerebrovascular measures of interest, all measured on MRI. Microbleeds were assessed by visual read, and we dichotomized it into absent or present, and also examined the location and dichotomized it into lobar or non-lobar locations. For white matter lesion volume, we used a continuous measure of global white matter lesion volume in the brain. And we also used a stroke-related characteristic variable, which basically included either lacunes, infarcts, or hemorrhages. So we grouped them all together. And the reason for this was because they all individually had a very low prevalence. So we grouped them together, and then was also assessed by visual read as positive, present, or absent. So our aim was to investigate this interaction effect between baseline cerebrovascular burden and baseline amyloid pathology on trajectories of tau. We performed linear mixed effects models. The formula can be seen here on the slide. And we focused on two main fixed effects of interest: the interaction between vascular burden and amyloid pathology, which reflected the effect on cross-sectional tau load, and then the interaction with, additionally with time, which reflected the effect on longitudinal tau accumulation. And then the models were performed separately for unimpaired and impaired individuals. So here you can see a quick overview of the cohort and the number of participants in each group. I think it's all quite according to expectation. Just one thing to highlight is that the amyloid positive cases are older than the amyloid negative cases at the same cognitive stage. And we know, of course, that vascular burden increases with older age, so this is just something to keep in mind. What we first did is to have a closer look at the presence of cerebrovascular pathology in each group. So here you can see microbleeds, white matter lesion volume, microbleed location, and the stroke-related characteristics in each of the groups. And when we then compared them to each other, we found that for microbleeds at the same cognitive stage, amyloid-positive individuals had more microbleeds than amyloid-negative individuals, but also cognitively impaired individuals had more microbleeds than unimpaired individuals. And this was mainly related to lobar microbleeds. For white matter lesion volume, we actually did not find any differences between amyloid-positive and amyloid-negative participants at the same cognitive stage, but we did find that impaired individuals had more white matter lesions than unimpaired individuals, which was irrespective of the presence of amyloid pathology. There were no differences for the stroke-related events between the groups. So on to our main results. Here you can see the interaction effects between vascular burden and amyloid pathology on baseline tau PET load on the top row and on longitudinal tau PET accumulation in the bottom row. And I will guide you through them per vascular factor. So for microbleeds, we observed a significant positive interaction effect with amyloid pathology on both baseline and longitudinal tau, but only in unimpaired individuals. So what you can see is that, for example, for the baseline effect, temporal tau is shown on the y-axis, tau PET SUVR, and then the presence or absence of microbleeds is shown on the x-axis, and the effects are colored for amyloid status. So individuals that were both amyloid positive and had one or more microbleeds showed the highest tau burden at baseline, but only in the unimpaired group. And also shown in the bottom for the solid green line, individuals that were both amyloid positive and had microbleeds showed the steepest increases in tau over time. For white matter lesions, we also observed a significant interaction effect with amyloid on baseline tau in unimpaired individuals, but we also corrected this effect for microbleeds since they were also related to each other, and this then did not survive correcting for microbleeds, suggesting that this effect was not independent of the effect we did find for microbleeds. And again, we did not find any effects in the impaired group. Neither did we find effects on longitudinal tau PET accumulation. For stroke-related events, we did not find any effects on baseline tau PET load, but we did find a significant interaction effect with amyloid on longitudinal tau in unimpaired individuals, but this was in the opposite direction, such that individuals with amyloid pathology and stroke-related events actually showed attenuated tau PET uptake over time. Again indicated by the solid green line, and I will come back to this in the discussion. So we then, so the key finding that we took from this was that the co-presence of microbleeds and amyloid pathology was associated with greater cross-sectional tau load and steeper increases in tau over time in specifically unimpaired individuals. So we then wanted to see whether we would find similar effects in an independent neuropathology cohort. And to this end, we included 1,610 cases from an Arizona-based neuropathology cohort who had data available on tau tangle density and amyloid plaque density, as well as CAA, white matter rarefaction, and infarcts. We performed similar analysis using linear regressions, and we observed very similar results, such that individuals that were both amyloid positive and had CAA showed the largest or the highest tau tangle density. And for infarct, we again observed this negative interaction effect, such that individuals who were amyloid positive and had infarcts actually showed somewhat lower tau in their brain. So these findings replicated the findings from the in vivo cohorts. So in summary, we observed that in the presence of amyloid, cerebrovascular pathology can modify tau accumulation in early disease stages. But we did observe differential effects on tau depending on the vascular factor, which may also explain some of the different or the mixed results that are currently seen in the literature. So what we saw was that for microbleeds, there was a positive interaction effect on tau, but actually for stroke-related characteristics, there was a negative interaction on tau. It could be that in our cohort, in the Biofinder 2 cohort, there could be some sort of survival bias in the sense that cognitively unimpaired individuals that have both stroke-related events and are amyloid positive are actually less likely to remain cognitively unimpaired. So that could be. We did not find an independent interaction effect of white matter lesions and amyloid on tau pathology, but we did find that white matter lesions in general were more related to cognitive impairments. So the effects we observed were specifically observed in unimpaired individuals, and it could be that additionally targeting microbleeds in these early stages may attenuate the rate of early tau accumulation. So I would like to thank my colleagues from the Swedish Biofinder study and Alzheimer Nederland for funding a 6-month fellowship with them. Also my colleagues from the Alzheimer Center Amsterdam, and happy to answer questions at the end of the session. Thank you.
It's my pleasure to introduce our next speaker, Prashanti Vermuri, who's going to be talking to us about white matter changes with cerebrovascular disease and Alzheimer's disease pathologies.
Thank you, Tessa. It's a pleasure to be here and to talk about the recent work that we have done on white matter changes with cerebrovascular disease and Alzheimer's disease pathologies. Nothing to disclose. The data that I'm going to be showing today is from Mayo Clinic study of aging. It's a population-based cohort where individuals are randomly sampled from the population and invited to be part of the study. There's no exclusionary criteria based on the pathology or vascular risk burden that they have. The data that I'm particularly going to show is from 1,500 individuals with PET as well as MRI imaging, and they have extensive imaging available, including a diffusion tensor imaging data. And the specific hypothesis that we have set out to test recently has been that measuring regional white matter dysfunction informs us about dementia etiology. And we believe that this information will be useful beyond the traditional biomarkers that are utilized for cerebrovascular disease and Alzheimer's disease. For example, for cerebrovascular disease, we utilize white matter hyperintensities commonly. And then a beautiful plenary couple of days ago that really convinced us that tau PET is the way to go to assess disease progression when you're trying to study Alzheimer's disease. So a question that I often get asked in this meeting is, do we really need a cerebrovascular disease biomarker? Treat amyloid and everything will be all right, because most of the trials exclude people with vascular disease, high vascular risk, as well as high vascular disease burden. But we forget a really important point, which is that cerebrovascular disease is more common than you know. In the general population, the prevalence of vascular contribution to impairment and dementia is very high, and to be able to measure that accurately as more therapeutics are available for dementia is really important, and it'll help us translate and confirm the findings that we see. And some of the efforts in the field in the last 5 years have been tremendous in trying to bring together various groups and try to understand what measurements do we make of cerebrovascular disease. And the focus has been on white matter, because white matter has about 1/8 of the blood supply to the brain compared to the gray matter. And you see that there are subtle changes in white matter even before the formation of lesions much later in the disease process. And measuring these changes is very useful for assessing the amount of cerebrovascular disease. An initiative by NINDS is trying to use diffusion MRI to come up with imaging kits. The study is called as MARC-VCID. It's a large consortium. And 2 biomarkers that they utilize are PSMD and free water that measure these early diffusion-based changes, and they're focused mainly on diffusion MRI. So why would we really need regional biomarkers when we have global biomarkers? PSMD is a measure that measures the extent of mean diffusion of water along the white matter skeleton. So the measurements are made let me try to say, yeah. Measurements are made along the white matter skeleton, and free water tells you the amount of diffused free water in the brain after accounting for tissue fraction. And the focus on white matter is because it's a very early change. When we have tried to look at regional changes in the MarkVCID kit, so we downloaded the MarkVCID kits and we wrote scripts in-house to try and understand where in the brain and where in the white matter does diffusion change if you have vascular changes 5 years before you see the diffusion changes. And we find that anterior white matter is much more vulnerable to damage compared to anywhere else in the brain. So I'm going to try and make a case that measuring genu of the corpus callosum is a really good target for trying to understand cerebrovascular disease changes, specifically early in the disease process. And there's significant biological evidence. I have to tell you that if you look at the pathology literature, they always are much ahead than the imaging literature is. And 20 years ago, there's been evidence that there's greater sclerotic changes in the frontal lobe arteries in vascular dementia compared to Alzheimer's disease. And given that genu of the corpus callosum is a late myelinating fiber which has very thin diameter, it makes it very vulnerable to vascular disease-related damage than anywhere else in the brain. We've focused on genu in the last 2 or 3 years, and we have got evidence from multiple sources to try and understand why it would be a good target for measuring cerebrovascular disease changes. And we find that when you look at pathology of the genu and you try to make measurements of cerebrovascular disease damage throughout the brain, it appears that measurements that you make in the genu at pathology maps on really well to the pathological burden that you see throughout the brain. It performs better than global diffusion measures as well as the traditional biomarkers that we've heard about, white matter hyperintensities, microbleeds, and infarctions, making it a really good target to measure. It is useful for predicting cognitive decline in the population, both cognitively unimpaired and mild cognitive impaired populations. And also, we have AI studies confirming that all white matter is not equal. So if you measure white matter damage throughout the brain, that might not be appropriate when you're trying to focus on measuring early changes due to cerebrovascular disease. We also followed it up with a causal modeling study because the focus right now is on lifestyle interventions, such as FINGER and the US Pointer study, where the intensive cardiovascular interventions seem to have an impact on risk of dementia. So we're trying to understand if we can use this biomarker to try and measure early changes that we see when we're targeting intensive blood pressure control. And what we find is that midlife modifiers, such as physical activity, individuals who are working out more, seem to have less risk of hypertension, and that contributes to much healthier anterior white matter. And if individuals have healthier anterior white matter, especially genu of the corpus callosum, they're at much lower risk of getting white matter hyperintensity lesions in the future, providing further evidence that genu-FA is a good early VCID marker. So the take home from this talk is that measuring white matter changes regionally, especially genu of the corpus callosum, would be an early CVD biomarker. Next, we move on to an AD white matter biomarker. Why do we need a Alzheimer's disease white matter biomarker, you may ask? We have tau PET. We have excellent biomarkers out there. We really do not need to measure white matter changes. Do they really matter as disease progresses? So we set out to test this hypothesis, and JQTN published a paper recently. He's a PhD candidate. And the data basically shows that if you utilize tau deposition, and you assess the amount of tau PET SUVR in the BRCA1, 3, 4, 5, 6 regions in individuals who are along the Alzheimer's disease spectrum. There are about 70 Alzheimer's disease participants in this study. You find that as the tau increases, as you go from BRCA1 to 3, 4 to 5, 6, you see that the extent of white matter damage changes. Initially, you see more damage in the uncinate, the hippocampal region. Slowly, you start seeing changes in the cingulate gyrus from the hippocampus. And as the disease progresses, as Alzheimer's disease marches on, there's a stepwise spatial changes in the extent of white matter impacted. So what? So why should we still measure? We understand that the whole brain is impacted, both gray matter and white matter. There's significant atrophy throughout the brain. So why should we measure it? And I'm hoping that these numbers would convince you. If you had BRAC ROIs and put them all together and try to explain the amount of variability in cognition, you can explain about 29% of variability in MMSE. With white matter integrity, you could explain about 31% variability, saying that it does give information. And if you put the entire information together, you explain the greater extent of damage and the greater extent of impact that Alzheimer's disease pathologies have on cognition. So the take-home number 2 is that measuring posterior white matter changes using diffusion MRI can provide complementary non-overlapping information to tau PET data. To summarize what I talked about, there is evidence that there's a differential mapping between dementia etiology and white matter health. Measuring white matter is important, but where you measure it, especially when you're trying to study late onset sporadic AD, is very important. And trying to distinguish between posterior and anterior white matter is important. The second point I wanted to make is we focus a lot on Alzheimer's disease, amyloid and tau PET biomarkers and plasma markers, but we tend to forget that the way in which AD pathology impacts the brain is through multifactorial pathways. And so to really do a good job with our modeling and prediction of someone's future risk of cognitive impairment, we need to consider the multifactorial pathways through which dementia etiologies are impacting cognitive decline. There's several new steps that we're excited about. We're trying to develop better biophysical models that can pinpoint to the mechanisms through which these etiologies impact brain health, specifically white matter as well as gray matter. GU has a great poster showing that NODI is a very good biophysical model compared to diffusion tensor imaging. And this work wouldn't have been possible without the superstars, the co-authors, the patients who make these studies possible, the funding agencies that allow us to publish and do this work. Thank you for your time.
So our next speaker is attending the session virtually, Mustafa Bourara, and they will be presenting on good plumbing for a healthy brain, new insights into the relationship between cerebral blood flow and tissue microstructural integrity.
Hello, everyone. These are the members of my group here at the NIA, and of course, all the credits goes to them. The research focus of my group is really centered on the development of MRI techniques and analysis to probe cerebral microstructure function and composition with high sensitivity and specificity. And the goal is to understand how specific determinants of cerebral tissue as measured using those imaging biomarkers impact hallmarks of aging and dementia, including cognition and physical dysfunction. Also, in my group, we are interested in understanding how modifiable and non-modifiable risk factors impact cerebral tissue as measured using those imaging biomarkers and also in combination with the other fluid biomarkers. But today I will focus my talk on our extensive work investigating the potential association between cerebral blood flow, or CBF, and cerebral tissue integrity, including myelin integrity in normative aging. Although the vascular dysfunction has been implicated as a potential mechanism for age-related neuronal tissue deterioration, the study of the direct relationship between vascular health and cerebral tissue integrity has received very little attention. So I have nothing to disclose. This research is motivated by our hypothesis suggesting that since cerebral tissue maintenance, including myelin synthesis, is an energy-demanding process, this metabolism becomes particularly sensitive to hypoxia and hypoperfusion. So in other words, with lack of nutrients, oxygen, and glucose support, it becomes difficult to support maintenance of cerebral tissue, including myelin synthesis. And also adequate cerebral blood flow is also needed for brain clearance from metabolic byproducts. So an analogy that I always like to say is we can see the brain like a house, And for normal functioning of a house, we need clear water coming in and also getting rid of the bad water. And you can imagine that any dysfunction on one of those processes might lead to some disastrous consequences. So I'm not going to go into details here, but to measure cerebral blood flow, we use the ESL technique. And this technique, based on some physics and mathematics, give you the cerebral blood flow map. But as you can see here, those maps are very noisy and hard to explore. So to address this issue, we developed a new post-processing filter that, unlike all other advanced filters, uses the non-local information by averaging voxels that are not necessarily close in distance to the voxel or to the index voxel. And those similar voxels are defined as those that exhibit similar trends or signal trend in the multispectral images, which are here in this specific case, the proton density, the control, and the labeled images. So we named this filter the NESMA filter, or the non-local estimation of multispectral magnitudes. We implemented all available filters for cerebral blood flow mappings, and we can clearly see here that the NESMA outperform all existing filters, including in terms of low bias and low dispersion, allowing us to have now high-quality cerebral blood flow mapping. Just for disclosures, this is the real NESMA, who is my wife. So now to probe cerebral microstructure, there are several techniques or a few established techniques, including diffusion tensor imaging, magnetization transfer ratio, relaxation times. All those techniques have helped us to advance our understanding of cerebral brain maturation and also neurodegeneration. But although they are sensitive to capture those microstructural changes, they are not specific. And due to the fact that the NMR, or the nuclear magnetic resonance signal in those techniques, it's a sum of the signal coming from different water compartments. And we know that cerebral tissue, it's a complex media, including white matter, which is compartmentalized, where we can account at least 3 different water compartments, including the myelin water, the intracellular water, and extracellular water. So you can imagine that any change or combination of changes here due to demyelination, inflammation, astrogliosis, and so on, although will be captured by those techniques will fail to tell you where the change is coming from, which hampers our capability for targeted interventions. So to address this specificity issue, in my lab we use, which is called multi-component analysis, including multi-component relaxometry and multi-component diffusion. And what we are trying to do is simply to isolate the fraction of the water coming from each of those compartments, because those compartments exhibit different NMR properties, including relaxation time and diffusion coefficient. And by doing so, we can now derive new metrics, including the myelin water fraction, which is a direct measure of myelin content in white matter, and also the norite density index, which is a direct measure of axonal density in white matter. But for lack of time, I will focus my talk today on myelination. So very briefly, we introduce a method called the BMC-MAC-DESPOT, which allow us to probe the myelin water fraction by combining images obtained using steady-state imaging, including SPGR and BSSFP at different flip angles, and then we fit our model. So after establishing the analytic expression to describe the signal of those sequences in a multi-component system, you end up having this high-dimensional problem to solve, which makes parameter determination, including the myelin water fraction, very unstable. So we stabilize the system using our Bayesian analysis. And if you are interested in the technical aspect, please reach out to us and we'll be happy to discuss that. So here I'm just showing those myelin water fraction map that I've used in BMC-MAC-DESPOT as function of age, and we can see the patterns of myelination increasing until middle age and then decreasing at later ages. We can now, thanks to the high spatial resolution of our method, we can look at small structures or complex structures that are very important in aging and also in neurodegeneration, including the brainstem. I'm just showing here example of papers where we apply the BMC-MAC-DESPOT for myelin quantification and to understand how myelination is implicated in cognitive impairment and also in physical impairment, and how different metabolic, vascular, and also other risk factors impact myelination. So please have a look, and if you are interested, reach out and we will be happy to help in implementing this method. So now that we can measure cerebral blood flow in myelin, we look at their association, clearly showing that participants with low cerebral blood flow exhibit low myelin content, and this association was significant in most brain regions investigated, providing evidence of the implication of deficit in cerebral blood flow in deterioration of white matter, and specifically in myelin integrity. So we push the analysis a little further. With collaborators, we look at the CARDIA study, which is a longitudinal and large study of adults to understand the vascular risk factors, and specifically look at the association between cerebral blood flow at baseline and changes over time in fractional anisotropy and mean diffusivity, which are metrics of the DTI analysis. Clearly showing here that participants with low cerebral blood flow at baseline exhibit steeper decline in fractional anisotropy and rapid increase in mean diffusivity. In other words, rapid decline in cerebral tissue microstructure. Interestingly, this association was mostly significant in white matter region, providing kind of support to our previous study and our hypothesis that since oligodendrocyte metabolism could be affected by cerebral blood flow and then may lead to this deficit in myelin synthesis. Now, the natural question will be why we should care about myelination. Well, for the simple reason is that here we clearly show the recent studies that participants with low cerebral myelination exhibit steeper declines in several cognitive domains, including executive function, processing speed, and memory, as compared to participants here in green that have high myelin content at baseline. Same thing with gait speed, which is an integrative measure of motor function. Participants with low myelination exhibit steeper decline over time in gait speed as compared to participants with high myelin content at baseline. And deficits in cognition and physical functions are hallmarks of aging and also of dementia. So in conclusion, we address central questions in the relationship between brain tissue microstructure integrity, specifically here myelin, which we believe with cerebral blood flow are key aspects in neurodegeneration and possibly targets for interventions, including through myelin enforcement, and this is people from multiple sclerosis knows how to do that, or also through cerebral blood flow modulation through lifestyle changes or interventions, and also shows how MR physics can address some questions that cannot be answered otherwise and motivate further investigations. Thank you very much.
So the last speaker for our session is Sindhuja Govindarajan, and she'll be talking to us about genetic variants underlying the neuroanatomical signatures of hypertension measured on structural MRI.
Thank you. Thank you for the introduction and the opportunity to wrap up this excellent session on vascular risk factors and imaging signatures. I personally have no disclosures. Now, we know without a doubt that the presence of vascular risk factors increases the risk for dementia, and that certain risk factors change the brain in ways that are measurable on MRI. With T1-weighted structural MRI, we can measure atrophy of gray and white matter regions, and with T2-weighted FLAIR images, we are able to measure the increasing white matter hyperintensities in response to cardiovascular risk factors. Now, what's important to note is that not all risk factors are the same, and not all age of onsets are the same. There's a greater risk of dementia with middle-age onset of hypertension, and this risk decreases with age. Now, with these two pieces of information, let's do something unconventional and have a pop quiz. Here, I'm showing you 8 subjects, 8 participants, roughly between the ages of 55 to 62. They're a mix of male and female participants, and they present with different combinations of comorbid cardiovascular risk factors. With this in mind, are you able to speculate what their brain can look like? Or even better, I'm showing you their gray matter volumes on the top and white matter hyperintensity at the bottom. And I want to see if you can guess which 4 of the participants from the left have their images shown on the right. Now I'll pause for some dramatic effect. Okay, I think that's good. So all 4 participants have hypertension, and all 4 participants had high BMI. And 2 of the participants had diabetes, and 1 each had high cholesterol or they were smoking. If you got the answers right, even 2 out of 4, please talk to me and I'd like to collaborate. But the reason I want to talk about this is because from group level observations, we get great information. We saw excellent biomarkers, their associations and how they are associated with dementia, both from cross-sectional and longitudinal studies. But can we move further and look at what's causing the heterogeneity among individuals? And can we dissect these imaging signatures to see what's coming from a certain risk factor? Because more often than not, they're comorbid. Why is it important? As we saw, the midlife onsets are contributing to higher risk for dementia. And in order to have appropriate interventions, we have subject selections that could potentially benefit from knowing what exactly is causing the heterogeneity and which participants are vulnerable to one risk factor related changes versus another risk factor. This is what we set out to do. We want to look at structural images and get the benefit of a single summary measure that can tell us something about a single specific cardiovascular risk factor. So we derived spatial patterns for recognizing abnormality, or SPARE scores. And that was aim 1. And the aim 2 was to understand what are the genetic variants that could contribute to either resilience to the SPARE changes or add additional risk. So I used the eye staging study. The eye staging study has imaging data from over 65,000 participants. But for this investigation, I chose the cognitively unimpaired participants between the ages of 45 and 85, resulting in a total training data of 20,000. We dichotomized hypertension presence using self-reported measures across the studies, as well as using the systolic and diastolic blood pressure values, resulting in those training data set volumes. Looking at the distribution of the different factors in participants, there were more men who were positive for hypertension than women. With race and education, the distributions were comparable. Structural MRI was processed using methods we've outlined in previous papers. T1 images were segmented using a multi-atlas segmentation method and harmonized so that effects of site variability, so scanner effects and sequence effects, are removed, and all volumes are brought to a space that's comparable across the studies. And volumes of white matter hyperintensities from FLAIR images were segmented using a deep learning method. With that, I performed a supervised machine learning method to separate control participants from hypertension participants. And from this, we're able to understand how much a single person's brain looks like it has the patterns of hypertension-related changes in the brain. And that allows us to place participants on this dimensional system where people with none of the effects are somewhere in the bottom corner with low scores, and people who express more of one risk factors patterns go along these further dimensions. And these are my results. Although my topic says hypertension, I want to show these other studies for comparison. So people with no risk factors are on the lowermost octant, and people with all 3 of the risk factors increased diagonally across. And people who had only hypertension moved only along the SPARE hypertension axis. The same was true for diabetes. They moved up along the diabetes axis, and obesity. And when these conditions were present together, as is common in the real world, the SPARE scores simultaneously increased for the comorbid risk factors. We wanted to make sure we're capturing what we find through group differences. So we looked at what's associated with these SPARE scores. We looked at the gray matter volumes and white matter hyperintensities. Now, even though there are large regions of overlap in gray matter atrophy across the different risk factors, the exact patterns of these changes were different between the different conditions. And as we saw in as we saw in Dr. Wimuri's talk, there's a lot of expression of frontal white matter across the cardiovascular risk factors, but hypertension had the highest association among all of them. Here I'm showing you a comparison with comparison of our marker with a conventional method of looking at the total volume of white matter hyperintensities. We're comparing the distributions of people who are normotensive, people who are stage 2 hypertensive, as defined by the criteria before, and people with stage 1 who were not considered stage 2, but they had higher than normal systolic and diastolic blood pressure values. On the left, you can see that there are differences in the peaks between the different distributions, but these differences are much more pronounced when we use our spare hypertension scores. We also wanted to investigate whether our spare hypertension is providing information that's the same as other markers we've already established. So we looked at spare AD, which is a marker for Alzheimer's disease calculated using a larger data set, including AD participants. And here on the right most is the marker for brain aging. So how old does a person's brain look compared to their chronological age? And as we saw in the US pointer study, the spare AD signatures are complement, or the spare hypertension signatures are complementary to the spare AD signatures. And I can hear you thinking, Sindhuja, you still just have group results. These are distributions everybody shows. What's so individual about this? Here we go. Do you remember the colorful indication of who was tested positive for what diagnoses? It's back here. The borders indicate that this person was positive, dichotomized as positive in that particular cardiovascular risk factor. Going through our images here, the first two participants had low scores on the spare hypertension index. Even though they had white matter lesions, they had higher spare obesity because in total, not looking at one or the other in isolation, in the total, the pattern looked like they had more of a spare obesity type of pattern, whereas these two participants had higher scores on spare hypertension. Now we want to know what's happening here. What could potentially drive the heterogeneity in the spare hypertension scores? So we did GWAS on UK Biobank MRI cohort with about 14,000 participants. We did an association after correcting for the principal components in genetics, age, sex, and intracranial volume. And what we found was there were the significant SNPs all in chromosome 2, which had a significant CAT score of greater than 12.37 and a beta less than 0, which means they were negatively correlated. So these SNPs also mapped to genes that were already cataloged in the GWAS catalog and associated with heart health and measures of gray matter and white matter structure, as well as to blood pressure and cholesterol. So spare hypertension lies in this, connects the dots of the triangle between the clinical features, the genetic features, and the imaging features. People with the variations on this particular, these SNPs it was a protective variant, so people had lower scores at older ages than the reference variant cohort. So in conclusion, we derived a marker that's more sensitive to hypertension than other markers. We found genes that were relevant, completing the triangle for spare hypertension. These can be used for personalized risk assessment and potentially be part of clinical trials for sample selection or in the future used in treatment management. Lots of future work. A subset of that is the association with spare hypertension at midlife and cognitive impairment at older ages in the I-staging cohort. Longitudinal changes of what is expected and what may be more at risk, who may be more at risk for cognitive decline, and to quantify the severity and treatment effects and explore the utility of this tool in clinical trials, which has been funded by the Alzheimer's Foundation, for which I'm very grateful. Having said that, thank you to all the co-authors and collaborators for the study, the different study contributors, and my PI, Dr. Christos Zavatsikos. Thank you.
Now we're happy to take any questions. Just come to the mic. Microphone 4.
Thank you for all of those talks. It was really excellent. I really appreciated that. I'm curious what you would think. I'm Jeremy Prutzen from Banner Alzheimer's Institute. I'm curious what you all would think or react to the idea about using white matter hyperintensities as a outcome of a trial, perhaps in younger persons, cognitively unimpaired population. Would there be some utility in that, or what are your thoughts about that?
So you don't see significant amount of white matter hyperintensity changes. Are you talking about diffusion MRI, or are you talking about white matter hyperintensity changes?
Volume.
Volume. It doesn't change much before 65. The dynamic range is very small. You don't see anything take off. But it depends on the population that you're trying to study. If it's early onset Alzheimer's disease, there's tau-related increase of hyperintensities. It's a non-specific marker. And if you're treating vascular disease, it's above 65 that you could use it as an outcome measure, but it doesn't change too much over more than a 2-year period would be perfect, but it depends on the question you're asking.
So I guess I could be a little more specific. So what if you randomize a group to a lower systolic blood pressure, for example, compared to a higher systolic blood pressure and track them over, 5 years or something?
There is data showing that it could be used. The Sprint Mind study had a JAMA Neurology paper showing that. They did use hyperintensity as an outcome.
And what about in, say, a 40-year-old or 45 to 55-year-olds? I know that paper started at about age 55.
Would be too early, but I don't know if anybody else agrees. Or others have comments from the audience.
Thank you.
Jasmine.
Hi there, it's Jazz Chatwall from Mass General in the Brigham. That was a wonderful session, thank you. I had a question. So we looked a little bit at spatial patterns and how those can inform etiology, but I was curious what the panel's thoughts were on using different white matter disruption measures. So say combining white matter hyperintensities, DTI, white matter hypointensities. Is there any utility in using different measures of white matter disruption together?
Yeah, we didn't. Can you hear me? Yeah, I think I cannot really answer the question in the sense that we don't know. We have seen that all of them give a different information. I think it's very interesting to understand what's the biological meaning behind all of them, but definitely possibilities to try to merge them in one index and then use them like all together for sure. Yeah.
Yeah, like using latent models like you did.
For example,
right.
using a latent variable model like we did, but for the radiological score.
Right. Mike 1.
This is Tim Hughes from Wake Forest School of Medicine. I'd like to congratulate you all on a great series of talks, especially how creative they were in many domains. For those that were using risk scores themselves for the vascular factors, I commend you for using the approach of actually breaking those risk factors apart. In particular, they represent multiple upstream vascular etiologies and pathways. In particular for hypertension, one of the tough things is it's often confounded by blood pressure treatment with antihypertensives. So if you can break out something and possibly look at the pulse pressure of an individual, that should be able to give you some indication. Have any of you looked at the pulse pressure in these?
No, but Pointer baseline enrollment has just wrapped up, so we have information about medication that we haven't integrated into this work yet, for example. And also there's a neurovascular ancillary study that is getting a lot of data that we can help use to understand these risk measures that are sort of more demographic in nature.
And maybe I can add, in our work, for the Framingham score, we actually looked at the single components, as you were mentioning, as a sort of supplementary descriptive analysis. It was very interesting. Indeed, hypertension was the one mostly contributing together, of course, with age and others. We did see something also with systolic blood pressure, but I seem to remember hypertension was the strongest one, yeah.
Great,
so we are reporting that, yeah. Thanks.
Microphone 3.
Hi. This is Clint Wright from NIDS. I just wanted to tell this audience that the Institute is very interested in anti-amyloid therapy in people with extensive vascular disease, people that might not have been included in some of the trials that we've been hearing about. So we have concept approval to run phase 3 trials on the safety and efficacy of anti-amyloid therapy in those groups. I will be here at the meeting if you want to talk about it further. Rebecca Hammer at NINDS or NIH.gov will be the program contact on that. Thank you.
Let's go to number 4.
Thank you very much. Christopher Chen from Singapore. So I'm just a neurologist, a stroke neurologist. So all your sort of wonderful new techniques, looking at different vascular parameters, can you reduce them to mechanisms? Do you think it's all driven by small vessel disease, large vessel disease, endothelial dysfunction? How do you relate that to that kind of treatable, druggable targets?
So,
I don't have an answer to that exact question of how can we how can we know the mechanism behind it yet, but that is the direction we're heading in. Especially with Luigi's work, you are working on the different types of vascular presentations, whereas I'm looking at a more summary level information across all the different types he's looking at. I think it's the direction we're heading at, and that's the best answer I have. Perhaps the person on Mustafa can answer that about the plumbing and the question asked. Oh,
we need to turn on his audio.
I have, I don't know if you can hear me, but
Yes, now we can.
that's a very loaded question, and I agree with you. This is what we are heading now. And disentangling all those mechanisms that are associated with each other is not that easy to do. But we are seeing now improved technology in terms of imaging and omics, and I believe this is in the upcoming years we will have some answers and try to understand and disentangle and dissociate those mechanisms and establish the causality link between them. But right now we cannot address that question, but it's a very good question.
Just to say that I'm really glad you're going that way, because we've made such terrible mistakes in the vascular field over the years, in that we've been so engaged in all the wonderful things that we can see, but we don't think of mechanisms to treat. So good luck.
Professor Shen, if I might add an analogy to the Alzheimer's disease field, 20 years ago, hippocampal atrophy was utilized as an Alzheimer's disease biomarker, right? 20 years ago, hippocampal atrophy was the end-all, be-all as a biomarker. Then we slowly discovered subfields of hippocampus are driven by different etiologies. We've been having a biomarker is helpful. That's what we're doing right now, coming up with a reasonable biomarker that overarching represents cerebrovascular disease. I think our next steps are what is driving which particular component of it. I think this is a great step forward. Just like hippocampal atrophy was a way to lead us in the right direction, these biomarkers actually will lead us down a path where we can figure out the mechanisms through which we can impact them.
Microphone 5 in the back.
Yes, hi. I'm Allison Rosen. I'm at Stanford in the Palo Alto VA, and very creative session. It was beautiful. This is in response to the SPARE, I'm echoing, the SPARE presentation. Beautiful work. One thing I would encourage you to do is to measure behavior and relate it to the regional distributions and look at the overlap, the shared regions. So for example, a lot of these disorders is associated with addiction, and to the extent obesity versus smoking differ, to the extent that depression regions and ventral impulsivity, look at behavior, because with the vascular risk, we can intervene in understanding the brain substrates and their relation to the behavior, reinforcing that would be very powerful, but beautiful work.
Thank you. I don't know if you expect a response.
No,
Oh,
just
thank you.
a comment.
I'd love to talk to you after the session. Thank you.
Happy to share.
And microphone 3, you've been waiting.
Hi, I'm Elijah from the United Kingdom. First of all, thank you for this wonderful talk. This is just a general question to the panel. So there's a lot of focus on using, the total white matter hyperintensities. I'm just wondering, for researchers, is it important to differentiate between deep versus periventricular white matter hyperintensities, and whether you have done that, and whether the results are robust, irrespective of the type of white matter hyperintensity in terms of the spatial location? Thank you.
The answer is yes. I'm jumping in. It is important, the location of the lesions, because as the other gentleman asked, it would tell us a little bit about the etiology of those presentations. I did not do this work, but Mohammed Habs from our group did this work. There's a 2018 paper. Please look it up. And he looks at the relative prevalence of frontal periventricular deep white matter lesions and how it affects brain changes, or brain aging.
Jan Johansson, Artery Therapeutics. Given ApoE's effect on hemorrhagic stroke, I didn't see very much of correlations of ApoE genotype to imaging. What's your comment about that? Or didn't you have access to ApoE genotype?
Exactly. It's an easy question for me to answer. We don't have it in Pointer yet. We're expecting it soon. So I couldn't look at that in my study, at least, but I don't know if anyone else wants to comment about ApoE.
Can I say something here?
Yes.
We have some studies, including from my group, where we are looking, for example, at the IPOE genotypes or isoform in association, for example, for myelination. And we know that the IPOE help in circulating lipids and transportation of cholesterol, which are the main constituent of myelin. And also there is some work that has been done also by Dr. Dean Douglas from the Wisconsin-Madison, also that look at the specific imaging biomarker, how they are modulated by the IPOE genotype. So there are some work that has been done in association with the neuroimaging. Thank you.
And a follow-up question. You are probably aware of that ApoE concentration in itself is a risk factor in the Copenhagen heart study. So if you have the information just on ApoE protein, that could be useful in itself. So the ApoE protein concentration in plasma.
Thank you. Yeah, I don't believe we have that yet either, but we can look into that. Thank you. And this is probably going to be our last question because we're at time. Thanks.
Well, thank you for letting me ask this question. My name is Narlin. I am at UBC in Vancouver, Canada. I work with exercise interventions, and we've been looking at myelin as an outcome using MRI. My question is, in the aspect of cerebrovascular disease, if you guys were to design a trial and try to target one element, what would you target? What would you think is our, brain plasticity equivalent for the white matter?
Mustafa, do you want to take the question?
Honestly, I don't understand the question. If you can reformulate it in a layman language, that would be nice.
Basically, if you design a trial, either lifestyle, drug trial, and you're looking at the brain, there's been a lot of trials targeting, for instance, gray matter volume for, exercise as an example, but there hasn't been a lot for the white matter. I understand white matter lesions might be one, but I'm wondering about new ideas for targeting in treatment, thinking about the white matter, not so much the gray matter.
Exactly. That's a very good question. And as unbiased as I am, I will target it, for example, myelination, like improving myelination through drug interventions, for example, or playing on the risk factor that may impact myelination or white matter integrity, but also there is axonal degeneration that also can be a therapeutic target. So that's the two elements that I will design if I had to do some such clinical trials.
All right, thank you. I hope the funders heard so we can apply for that.
Thank you, everyone, for coming. We had some great questions. We have questions online. We'll try to answer them. But it's been an excellent session, and thank you all for stopping by. Thank the presenters.